Oh, right, I already started recording. I don't know why. I feel like I haven't done this in a couple of weeks just because I was away last week, and then the week before that, I believe you guys had a midterm, so it's been a while, but oh yeah, but wait, the week before that was, last week was reading week, and the week before that I was away. That's why I feel like it's been a while since I've done a virtual session. But yeah, I hope you guys are doing well. I think we're just gonna get started now. So can you guys see my screen? You can just send me something like in like a yes or a no, like in Teams. You don't have to turn on your mic if you don't want to. Okay, perfect. Awesome. So yeah, welcome to tonight's session. It's the same one that I did on Tuesday, and this covers lectures 20 to 22. So 22, I believe you did on Monday. And then I'll also be answering any questions that you may have. So lecture 20 was all about exercise metabolism. Sorry, this may be, I don't think this was the one that I used last time. Sorry, I think I opened the wrong slide. I just have so many files on my computer that seems like that's what happened. Sorry, I'm going to stop sharing my screen just to be sure that I have the right version of this, because I did one last year as well. Okay, so I think this is the right one. This is the updated version. I was just checking to make sure that the dates were right, but yeah, sorry about that. So yeah, we'll be doing a review of lectures 20 to 22. And then of course I'll be available to answer any sort of questions that you may have, as well as throughout the session. So lecture 20 was about exercise metabolism. So exercise has to do with, everyone already knows what exercise is, but I guess like before I took this class, I didn't really realize the extent of like what happens when you exercise and where you get the energy to exercise from. So this is kind of what this talks about. So the first place that you tap into when you need an energy surge in order to perform some sort of exercise would be the phosphocreatine system. And so from this system, you would get something called creatine phosphate. And then we'll talk about later what happens specifically, but essentially you're going to use that phosphate to perform substrate level phosphorylation and then create ATP. The second energy system that you would tap into once that phosphocreatine system runs out is the lactate system. So in this case, you would be going through glycolysis and then fermentation. And then the third system that you're going to tap into is aerobic respiration. So here you would be performing glycolysis, then the Krebs cycle, and then the mitochondrial electron transport chain. So this is the stuff that we talked about before with, the 3 steps in it and everything. This is the ideal version. This is the one where if you're not exerting a lot of energy and you don't need an instant surge in energy like you would for exercise, you would always be performing aerobic respiration, which of course requires a sufficient amount of oxygen. However, when you're exercising, oftentimes you don't have as much oxygen available to you just because of the nature of the exercise itself. So you need to use anaerobic methods in order to extract that energy. So the first system that I was talking about before was the phosphocreatine system, and this is great for the initial power usage, and it starts up very quickly, but it also runs out very quickly. So it runs out within around 10 seconds of starting it. So it's only like a very temporary storage of energy, after which you have to move on to the next system, which would be the lactate system. So once it runs out, you don't have any more creatine phosphate, and that means that the process stops. So in this case, it's a substrate level phosphorylation, just like what we talked about with aerobic respiration. So we said that for glycolysis in the Krebs cycle, although the main point is not to make ATP, you make a little bit of ATP. And so you would make that through substrate level phosphorylation, right? Because you're just directly adding on a phosphate to ADP to create ATP. So that's what that is. So that's why you don't make like a ton of ATP when you do this, but you still make enough to keep you going for your exercise purposes. And it's a good temporary solution solution. And of course, the reason why this system can't last forever, or at least one of the reasons why, is because the creatine storage that you have in your muscle cells runs out at some point. And in this process, it's an anaerobic process, as I said before. Oxygen is not being used. And as I said prior, one of the main reasons why oxygen is not used and why you need to use anaerobic processes to get energy when you're exercising is because you're not breathing enough to be able to sustain the other ideal process, which is aerobic respiration. We said before, just as a review, that glycolysis is an anaerobic process, and but the thing is that the Krebs cycle and the electron transport chain are aerobic. And so that's why you would still need oxygen present, and you would need a lot of it as well. And remember that the oxygen, especially, it comes in the others in the Krebs cycle as well, but it's also the terminal electron acceptor of the mitochondrial electron transport chain as well. I'm also like, I kind of have a feeling that I'm doing the wrong slides because I did a different session on Tuesday, and this is the one that I uploaded before reading week. Do you guys remember if you guys, I feel like we already covered this with you guys, I'm pretty sure. I think I went over this slide. It's so hard to remember now because of reading week, but I'm pretty sure, does this sound like old to you? Does this sound like something we've done before? Because I feel like I've gone through this with you guys.
This is my first time.
Okay, yeah.
So I don't know what you've gone over.
I do feel like, I mean, I definitely went through something else, and I think that this is something I did during reading week. So I believe, yeah, I believe that, I haven't been to many of your sessions, so I wouldn't know. Yeah, no worries at all. I definitely did this before reading week as well, but I don't know if it's just because you guys didn't do any new sessions, so, or any new lectures except for the Monday one, so that might be why. But I'm just gonna take a look just to make sure that what I have uploaded on D2L is the same one, because my computer crashed this morning and I don't know if it gave me a different version of my documents that I had from last year. I need to get a new computer for sure. Sorry about that, just bear with me for a second. And now it's frozen. Okay, I think this is the wrong version of the slides as compared to what I had before. I don't know why my laptop never cooperates with me, but I think I still have a backup file of my real ones that I think got, it didn't like auto save this morning. That might be why. Oh my goodness. I'm so sorry, guys. I literally can't find where my file went, even though I literally used it on Sunday or on Tuesday. This is so frustrating. Okay, I think this is the newer version because I found this one in my recovered files, so this one might be the better version that I should be using, but it's almost exactly the same. I just looked over the slides before I started, and some of the last ones didn't save well, so I think I got the backup file. It should be fine. Yeah, so sorry about that. This is why I think I probably should replace my computer sometime soon. Right. So yep, so we talked about the phosphocreatine system. Again, this is the first one that we go to use. It gives us a good temporary amount of energy using that phosphate that we cleave off of the creatine. Then we can perform substrate level phosphorylation, which is where we add that phosphate that we just got from the creatine, and we add it on to ADP to create ATP. And so this is an anaerobic process because it doesn't require any sort of energy. And the way that the phosphate is cleaved off of the creatine is through some sort of enzyme that you don't need to know. And again, that all of this is stored in the muscle cell, so it makes for very easy access when you first begin exercising. But of course, when this runs out, you need to move on to the next system. And in this case, that system would be the lactate system or the lactic acid system. And so this is our second course of action that we would take. I already talked about the two different kinds of fermentation systems. So I talked about lactic acid fermentation, and I talked about alcohol fermentation, but I did that in the session that I did prior to reading week. And so if you want the slides for that, they're already posted on D2L under my section. But yeah, so basically we're just going to talk about the lactic acid system. Just a quick recap now. The ethanol system is one that you should definitely know. It's just that it doesn't apply to humans, which is why we're not talking about it in the context of exercise. But yeah, so with the lactate system, it doesn't last very long. It's only about a minute long, but it's the second mode of defense that we go to, not necessarily defense, but you know what I mean? Like the second resource that we would tap into to make ATP, at least in quantities that are sufficient for exercise. And it's also an anaerobic process, which is good because when you're exercising, as I said before, you're not really breathing too much. And so essentially, as I said, the week that I did this, the whole goal of fermentation, whether it's lactic acid fermentation or ethanol fermentation, is that you are trying to regenerate your supply of NAD+. So that seems really counterintuitive because if you're using this for exercise, why are you trying to regenerate NAD+ instead of trying to make ATP? Well, the thing is that with fermentation, the goal is to regenerate NAD+ because NAD+ is a key ingredient in the process of glycolysis, and glycolysis is the one that produces ATP. So essentially what we're doing is we're supplying the cell with the necessary reactants to undergo glycolysis because that's going to give us the ATP that we need. So what happens really is that you have your cell undergoing glycolysis normally, as always, right? And we talked about glycolysis a few weeks ago now, but the slides are up on D2L. In case you have any questions, just let me know. But yeah, so the cell can undergo glycolysis in the absence of oxygen as well because it's an anaerobic process. But one of the necessary ingredients for this would be NAD+, which are those empty suitcases where we would be adding electrons in to fill them up. So it's the mobile electron carrier. And we get those electrons when we oxidize the glucose that we start out with to create the pyruvate, right? And so when we oxidize something, we're taking electrons away from it, and we have to add those electrons somewhere. They don't just get to disappear into space. So what we would do is we would grab those electrons off of glucose and add them to this empty suitcase. That's the way that I visualize it in my head. It's called NAD+. And then when we fill that up, it becomes NADH, which is the reduced version. It's reduced because it's carrying lots of electrons now, and then we ship that off to the electron transport chain so that we can use those electrons to create that proton gradient, and then we can have that gradient dissipating through ATP synthase, and then we make ATP. That's the stuff that we learned about before. But in the absence of oxygen, we can't go through those two steps. We can't go through the Krebs cycle, and we can't go through the electron transport chain. So what we have to do instead is we have to basically just keep going through glycolysis over and over and over and over and over again. And as we know from before, glycolysis doesn't produce that much ATP because of the substrate level phosphorylation. So you only produce about 2 ATP for every glucose molecule that you start out with. It's not a lot and it's not ideal, but if you're exercising and you don't have enough oxygen for the electron transport chain, then it's probably better to make a little bit of ATP than to make none at all, right? So essentially with fermentation, it starts out with glycolysis happening the way it normally would. And then after a while, glycolysis happens and you run out of NAD+. So glycolysis can't happen without that. So our goal here is to regenerate our supply of NAD+. So what we would be doing is basically after glycolysis, you end up with the product pyruvate, right? We said before that for each glucose molecule, you produce 2 pyruvate molecules during glycolysis. So with this pyruvate, what we do to that is we then convert it to lactate. So we convert the pyruvate to a molecule called lactate. And the way that we do this is by reducing the pyruvate, meaning we add electrons to it. So we're adding those electrons, but where do we get them from? Well, we get them from the NADH that we produce during glycolysis. Because again, NADH is the one that's filled with electrons. It's the reduced version. So when we grab the electrons from NADH and we add them to pyruvate, we're effectively reducing the pyruvate into a molecule called lactate, and we're oxidizing the NADH back to NAD+. And then when we do this, NAD plus can then be used to kickstart the process of glycolysis over again, and then we can keep producing ATP. So that's kind of the goal of fermentation, is to regenerate one of the major reactants for glycolysis so that it can then produce ATP. Now, the problem with this system, other than the fact that it doesn't last for very long, is the fact that you produce lactate. And lactate is essentially just lactic acid. It means the same thing, but this is toxic to our bodies. And so an accumulation of lactic acid is toxic, and it makes it very hard to it gives you like that soreness sensation that you would get after some sort of strenuous exercise. So if you ever work out and then you notice that the next day or the next couple of days you feel really sore, it's because of that lactic acid buildup. And the reason why you breathe very heavily after very strenuous exercise is because when you breathe, the oxygen that you inhale actually helps to eliminate that lactate from your body. So it's kind of like a double-edged sword. You're producing a lot of ATP, but at the same time, you're also producing a lot of toxins, and that accumulation of those toxins is very, it's not great. But of course, there are other ways to get rid of it. And exercise is very healthy because generally speaking, you're not going to produce enough for any sort of real toxic effects to happen to you other than just like that soreness that you would be getting. So yeah, just a key thing to remember is that with fermentation, you're not actually making any sort of ATP. You're making NAD plus because that's going to be used in glycolysis, and that's going to make ATP. So we're already restoring NAD plus, which is one of the key reactants for glycolysis. But what about glucose, right? Glucose is the main reactant for glycolysis. Basically, the whole goal of glycolysis is to oxidize glucose into pyruvate. So we need glucose to start. So where do we get that? Because chances are if you're working out, you're not also munching on a granola bar as you're lifting weights, right? So where do we get that glucose? Well, most of the time you would be getting that glucose from the liver, because glucose is stored in the liver in the form of a polysaccharide called glycogen. So a polysaccharide, just as a refresher, is basically a chain of sugars, right? So it's a chain, like multiple chain of sugars. And so this is essentially these glucose molecules that are linked together using glycosidic bonds. And this molecule is called glycogen. And so what we can do is we can chip away at the glucose that's stored in our glycogen reserves. This is typically stored in the muscle cells. And then when we run out of, and then basically when we're going to perform glycolysis and we need that first ingredient, glucose, we can just extract each individual glucose monomer from the glycogen polymer. And the way that these glucose monomers are cleaved off is using an enzyme, of course, and you don't need to know the name of this enzyme. And in fact, there's actually different enzymes that are used to cleave glycogen, just because there are different linkages in the glycogen. You can have branched ones and non-branched ones, and each one would have a different kind of enzyme that's specifically made to cleave that specific linkage. But of course, that's not something you need to know. So yeah, that's where we would be getting that. And then, of course, the third thing and the most ideal step, which you can only do if you have sufficient amounts of oxygen, would be aerobic respiration. So of course, this would be glycolysis, and then you would have the Krebs cycle, and then you would have oxidative phosphorylation. And it takes a lot longer for this to start, but it lasts for a really long time, something like hours. And so this is what we would be doing right now. Right now, as we speak, we're all going through aerobic or cellular respiration, right? And so this is how we produce the bulk of our ATP, using oxidative phosphorylation. And so something that we've learned is that you start out with glucose, right? We know that with glycolysis, you start with glucose, you make pyruvate, you go through the Krebs cycle, you make a bunch of those reduced electron carriers, and then you take them to the oxidative phosphorylation, which is the electron transport chain, and then you make that proton gradient, and then you dissipate the proton gradient and you make ATP. right? But the thing is that you don't necessarily have to start with glucose. There are different molecules that you can start out with where the body knows basically what to do to convert it into something that's usable, and it's going to fall into one of these steps. So, for example, carbohydrates and some amino acids, as well as glycerol, those are all going to come into play at some point during glycolysis. It doesn't necessarily have to be glucose. If you're working with fatty acids or some sort of amino acid, it's going to come in through that pyruvate oxidation step. And if you're wondering what that is, I don't know if you remember this, but do you remember when we talked about the cellular respiration? We said how between glycolysis and the Krebs cycle, there has to be an intermediate step where you convert the pyruvate that you made in glycolysis into something called acetyl-CoA, because acetyl-CoA is the entry molecule for the Krebs cycle. So what we're doing to the pyruvate to convert it to acetyl-CoA is oxidizing it. So that's what this step is here. It's just the step that links together glycolysis and the TCA cycle. And oftentimes, and I think your textbook does this too, people usually group this step as part of the Krebs cycle. So some people don't even distinguish it as its own separate thing. They just include that as part of the TCA cycle. And then if you start out with with other amino acids, there's lots of different ones. There's around 20 of them that you make in the body. Some of them can come in through the pyruvate oxidation step. Other ones will come in sometime through the TCA cycle. But in general, they all kind of lead to that same place with that same process. It's just that they need to be modified differently, and they can start at different steps. But the model one, the main one, is the glucose, which is the one that we talked about and that we learned about in detail. So that would be the end of that first lecture. Do you guys have any questions about anything that we talked about just now? I'm just going to check my phone because I think I have Teams installed here, and then that way you don't have to say anything on the screen. Oh, I just saw your message. Thank you for being so understanding about my annoying laptop situation. Right, so it doesn't look like there are any questions, so we're just going to move on to photosynthesis, which was lecture 21. So photosynthesis has quite a few steps. It's very similar. Okay, so it's a little bit similar in terms of concept to the electron transport chain, but it's also different, and because of the fact that obviously what you're producing is different and what you're starting out with is different. So that's one thing. Another thing is that sometimes it's easier to think of them as being opposites, like to think that the electron transport chain or cellular respiration is basically the opposite of photosynthesis. It's not, but it kind of like it makes it easier to understand if you think of them as opposites, but do not say that on your midterm because it's technically wrong. They're not opposites. Like factually speaking, it's just that the way I think of it is like by thinking of them as almost being opposites, it makes it easier for me to understand, but don't write that down. Just if it helps you understand it, then it helps you understand. Another thing would be obviously photosynthesis is something that happens in photosynthesizing plants and like algae and stuff like that, but it doesn't mean that cellular respiration doesn't happen at the same time. Plants are simultaneously undergoing cellular respiration and photosynthesis. So it's not like two things like you either have one or the other. Because cellular respiration produces different products than photosynthesis does. Photosynthesis produces sugars like glucose and oxygen, whereas cellular respiration, you're actually using oxygen and you're using the sugar and you're trying to produce ATP. So it's different, but they are kind of, they're very, if you understand the electron transport chain, this should be relatively easy to understand. And in fact, you don't go into as much detail with photosynthesis as you did with the electron transport chain, just because the goal was to help you understand the concept of things like that. As opposed to like as the concept of understanding like electron transport chains in general. So this is just another example of that, but you don't need to be as concerned about the details. So yeah, in terms of the equation itself, you start out with 6 carbon dioxides and 6 water molecules as your reagents, and your products would be 1 sugar, usually glucose. This is just the general formula for a lot of monosaccharides. And then you have the 6 molecules of diatomic oxygen. And so basically, one of the things that catalyzes this reaction is light, which would come from sunlight, or specifically photons from the sun. So photosynthesis, who undergoes photosynthesis? That would be plants, algae, and some bacteria. The goal of photosynthesis is to convert carbon dioxide and water into sugars, Sometimes it can be other organic materials as well, but usually it's some sort of sugar, usually glucose, and oxygen. And where does this happen? In these members, so plants, algae, and some bacteria, photosynthesis would happen in the chloroplasts, right? Obviously, um, with bacteria, they don't have membrane-bound organelles, so they don't have chloroplast, so it would be happening somewhere in their cell. But generally speaking, when we're talking about eukaryotes, photosynthesis would happen in their chloroplasts. So there are two parts to photosynthesis. So this is a little bit tricky, so I try to break it down very slowly, but please let me know if you want me to go back and explain something, or if there's something that you want me to explain differently. So there's two parts to photosynthesis. There's the light reactions, and then there's the dark reactions, which are also called the Calvin cycle, right? And so as you would imagine, the light reactions are the ones that require sunlight, and then the Calvin cycle or the dark reactions can happen at night as well. So oxygen and sugars are the only true products of photosynthesis, although in the light reactions you make intermediate products, and in the dark reactions you also make intermediate products. Those products are only used to start the next reaction. They never leave the chloroplast themselves, and you can't actually isolate them, right? So the only actual products that you're making are the sugars and the oxygen. You make the oxygen in the light reactions, and you make the sugars in the dark reactions, or the Calvin cycle. But for example, in the light reactions, you also make a little bit of ATP and some NADPH. But that's not an actual product because it goes straight to the Calvin cycle. It doesn't actually like leave the chloroplast or anything like that. Same with the Calvin cycle, you end up producing NADP plus, and ADP plus some inorganic phosphate, but that is not an actual product. Neither of those are, because they never actually leave the chloroplast. They're just made so that they can keep the next cycle going, right? So these just feed into each other, they never leave. These are the only true products, oxygen and sugars. So this is kind of the anatomy of a chloroplast. It's a membrane-bound organelle. It's a little bit similar to the mitochondria in the sense that it has a double membrane. So there's the inner and the outer membrane. Here you have each of these green discs are called a thylakoid. And a stack of these thylakoids are called granums. So this would be one granum, that's another, that's another, that's another. The anatomy of the chloroplast is pretty important, I would say, in this class, just because I know Dr. Durnford has like a particular interest in photosynthesis and in chloroplast. So it's nice to understand what the parts of it are. And so basically, if you're looking at the chloroplast from up here, you can see that there's a cluster of pigment molecules that are embedded in the membrane, and this would be the thylakoid membrane. So each of these discs is a thylakoid, so the membrane that surrounds it is the thylakoid membrane. This would be one individual chlorophyll pigment. And you can imagine that chlorophyll is green, which is why a lot of plants are green, leaves are green, things like that most of the time. And also why the thylakoids themselves are also green is because they're very highly concentrated in chlorophyll pigments. So chlorophyll kind of looks like this. It looks a little bit like a phospholipid, sort of, but it's definitely different. One of the commonalities is that it has the hydrocarbon tail, so similar to the phospholipid, but not quite. It also has a porphyrin ring, and this ring is the light absorbing head of the molecule. So again, it has a lot of chlorophyll pigments, as you can see here. They have chlorophyll B and chlorophyll A, depending on the identity of the plant, or the whatever it is that's undergoing photosynthesis. And it has this magnesium molecule in the center as well. And that helps with some of the chemical reactions that you don't really need to know very well in detail. If you take biochemistry, then you'll probably need to know this in greater detail. Biochemistry is a second-year course. It's really good, and I think it's required if you're doing a biochemistry degree or a biology degree, but yeah, you would need to know what the magnesium is for. So yeah, what about pigments, right? We said that chlorophyll was one example of a pigment and that it's green, but there are other kinds of pigments as well. So chlorophylls absorb blue and red light, and they reflect green light, so we see them as green. Because the thing about pigments is that the color that they absorb is not the color that we see them as, right? The color that they reflect is the color that we see them as. So with chlorophylls, they're absorbing blue and red light, but they reflect green light, which is why we know chlorophylls as being green. Same with carotenoids. They absorb blue light, but they reflect back orange light. And so because of that, we see them as being orange. So it's not the color that it's absorbing, it's the color that it's reflecting that we see. So yeah, back to the chloroplast, you have the thylakoid, you have the intermembrane space, very similar to the mitochondria in the sense that it has the outer and inner membrane, and the space between the two membranes is the intermembrane space, inter meaning between. You have your granum, which is your stack of thylakoids. The fluid of the chloroplast is called the stroma. So that's kind of like when we were saying that the fluid of the cytoplasm is called uh, the cytosol. The fluid of the mitochondria is called the matrix, and the fluid of the chloroplast is called the stroma. Right, so it's just a different name, and then you have so each of these individual discs is a thylakoid. Inside of the thylakoid, it's hollow. So these thylakoids, like the hollow parts on the inside, is called the lumen. Lumen in general in biology, you'll find that it just basically refers to some sort of like void, some sort of like hollow space. So the thylakoid lumen is the empty space inside of each of those little thylakoid discs. Right, so I think we're ready now that we understand the anatomy of the chloroplast. We understand the concept that there are two reactions essentially for photosynthesis. You have the light reactions and the dark ones, also known as the Calvin cycle. I think it's best that we get into each of the individual steps now. So the goal of photosynthesis, for step one, which are the light reactions, is to oxidize water to create molecular oxygen, and also to make ATP and NADPH so that we can transfer those products to the Calvin cycle. And those are byproducts. So those are not our actual products. The main product that we would get from this, the real one, would be oxygen. And then the location in which this happens is the thylakoid membrane. So that would be like just, the membrane of one of these discs. So this is a really helpful photosynthesis animation. I watched it a lot, and I think I might even go through it. Usually I don't go through my videos during my sessions just because they're just there to help, but I think this one makes it a lot more clear than than any of the diagrams that I could be using. So I'm just going to go through that really quickly with you. I have like a specific timestamp, but we're not going to go through all 4 minutes.
We will examine what happens with the photon striking photosystem 2 first.
Oh.
Each strike will channel the photons of light.
I don't know why it started away from the
The
beginning.
Calvin cycle of thylakoid is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems called photosystem 1 and photosystem 2 that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photon striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH.
Okay, so hopefully that was a good introduction to that, I usually, like, if I was in class, I might try to draw some of these things out, but I find that when we're just virtual, it's better to just show the video first. I did that last year and they seem to find it helpful. But of course, it's going to be linked and I'm going to put it on D2L as well, like the entire slideshow. So you should be able to find that if you ever want to watch it again, especially while you're studying. But yeah, we're just going to take a closer look at the light reactions that were talked about in the video. So essentially, this is kind of what you have, right? You have the thylakoid membrane. This side is the thylakoid lumen, so that was the empty space in the thylakoid discs, and this part was the stroma of the chloroplast. So that would be that the fluid, right? And so essentially you have between them a membrane, which is the thylakoid membrane. And on this membrane, you have embedded several different proteins, different complexes, very similar to with the mitochondrial electron transport chain, which that one had like the complexes 1 through 4, and then the 2 mobile electron carriers and stuff. But essentially, this is a little bit different just because the process itself is different. So starting over here, you have photosystem 2, but you also have one called photosystem 1. It's a little bit counterintuitive to think of it as going in this order, photosystem 2 and then 1, but that's just how it is. And so essentially with photosystem 2, you can see it's very green because it's very highly concentrated in chlorophyll pigments. And so what happens is you have essentially like a photon of light that hits photosystem 2. And that light, of course, comes from the sun. So you have the photons that are hitting photosystem 2, and it's kind of exciting the electrons in one of the chlorophyll pigments in here. So that excited electron gets passed around from chlorophyll to chlorophyll to chlorophyll, gets passed around, and then eventually it hits the P680. And the P680 is a special pair of chlorophyll pigments. They're just called P680. They're just, it's just a particular kind, and they're in the reaction center of photosystem 2. The reaction center is where the special pair of chlorophylls are located. And so they're special because they then pass this electron, the excited electron, over to the next carrier in the chain, which is the plastiquinone. So they pass that excited electron over to plastiquinone, and then when they do this, they also split water. So water splitting is essentially basically exactly what it sounds like. They have a water molecule because water is one of the reactants in photosynthesis, and they split it into atmospheric oxygen and a proton, right? So now you have a proton on this side of the membrane, which is in the lumen. So you're already making this more acidic, and that's just step 1. So now you have this excited electron at the plastiquinone complex, right? And because there's an excited electron here, then this plastiquinone becomes reduced. So instead of calling it PQ, we call it PQH2. It's not super important, but it's a little bit similar to when we talked about ubiquinone and how ubiquinone we usually call Q, but then when it's reduced, we call it I think we call it QH2. Yeah, we call it QH2. And then when it's oxidized again, so when it donates that electron that made it reduced in the first place, it comes back to its original oxidized form, which is Q. And in this case, with plastiquinone, once plastiquinone donates that excited electron over to the next complex, which is cytochrome B6F, then it's going to become back to its oxidized form, which is the PQ. And when it does this, it also pumps a proton to the thylakoid lumen. So it pumps it from the stroma to the lumen. The next thing that happens is now you have your excited electron that's been passed over to cytochrome B6F. So when this happens, cytochrome B6F has this excited electron, and really all that you need to know is that it passes it on to a different complex called plastocyanin. And this one's a lot more mobile. You can see it's like, and not really embedded in the membrane. It's more so attached to one side of the membrane facing the lumen. But plastocyanin takes that excited electron and then passes it over to plastocyanin takes that excited electron and passes it over to the other photosystem called photosystem 1. So photosystem 1, at the same time as photosystem 2, is being hit with light from the sun. And the reason why the sunlight and the photons are hitting it, so even though, like, obviously the sun's not just only pointing at photosystem 2 and only pointing at photosystem 1, but they're the ones that have the chlorophyll pigments that can be excited by the photons from the sun. The other two complexes don't have those pigments, so the sunlight doesn't affect them. Same with plastocyanin, so the other 3, right? So that's why it's just the photosystems 2 and 1 with their very high concentration of chlorophyll pigments. Those are the ones whose electrons can be excited by the sun. So while this whole process is happening, at the same time, plastocyanin is also being excited, right? So it's getting an electron that's being passed over. Sorry, photosystem 1 is also being excited, and it's getting that electron passed over from plastocyanin. And then that electron is going to be further excited, and it's going to be passed around from from pigment to pigment, all the way until it reaches the reaction center. And in the reaction center, you have another special pair of chlorophyll pigments called P700. So we said that the special pair of chlorophylls in photosystem 2 were P680. The special pair of chlorophylls in photosystem 1 are P700. That's just their names. The P is pigment. So P700 is going to then pass that electron over to NADPH. And then that NADPH is going to be that terminal electron acceptor, because this is the final destination of the electron. So it actually does this, I believe, twice. Once from the one of the electrons that's excited in the photosystem, and once from the electron that was brought over from this whole chain of reactions that happened. So when this happens, right, NADP is going to be reduced because it's accepting that electron from photosystem 1, and it's going to be reduced into something called NADPH. So this is very similar to NAD and NADH. It's just that it has that additional phosphate, which accounts for the P, and this is the electron carrier that's used in the process of photosynthesis. We talked about NADH as well as FADH2, but those had to do with cellular respiration. So they're all kind of the same idea. They're essentially NADP would be the empty suitcase, so carrying no electrons. NADPH would be the reduced version, so the one that's carrying electrons. It's just that the identity of the electron carrier itself is different because the process is different, right? So essentially, when photosystem 1 passes that electron over to NADP, NADP is going to be reduced to NADPH, and it's the terminal electron acceptor. So another key difference between photosynthesis and cellular respiration is that in that final step where complex 4 passes the electron, that electron onto its final destination, the final destination was oxygen, because oxygen is the terminal electron acceptor in the process of the mitochondrial electron transport chain. But with photosynthesis and the electron transport chain that's used in the light reactions, the terminal electron acceptor, or the final electron acceptor, is actually NADP. So the final destination to which that electron that's been bounced around goes to is NADP, and it's reduced to NADPH. So this is one of the intermediate products, not the final product, one of the intermediate products that are passed over to the Calvin cycle so that they can be used later. And then another thing, of course, would be, of course, the oxygen is the actual product, and then the NADPH is something that's passed on, as is the ATP. Because how do we make that ATP? Well, it's basically the same idea as what we would do to make ATP in the mitochondrial electron transport chain. So essentially, you have another molecule embedded in the membrane called ATP synthase. It's just a complex of proteins, and it's an enzyme. You can tell it's an enzyme because of the suffix ase, right? So ATP synthase uses proton gradients, and then it uses the energy, the electrochemical gradient held in that proton gradient. And when it dissipates that proton gradient, it can use the energy that it extracts to force together ADP and phosphate. Because to make ATP, you have to join phosphate with ADP, right? Because ATP has 3 phosphates, ADP has 2 phosphates. The thing is that this is a very unfavorable reaction, and it costs a lot of energy because phosphates are very highly negatively charged. And so with ATP, you have 3 of them in a row. You know that that's a lot of negative charges very close together, and you know that negative charges repel, like charges repel. So to be able to add a 3rd phosphate onto a molecule that already has 2 is a very unfavorable reaction that requires energy. And a great way for us to get this energy without having to expend too much to get it in the first place would be to use that proton gradient and use ATP synthase to force the ADP in the phosphate using the energy that it gets from dissipating the electrochemical gradient. So that's exactly what happens. And the thylakoid lumen side, after all of these electron transfers, right, we have a very acidic lumen. So it's acidic because it's rich in protons. The reason why it's rich in protons is, as I said before, when the electrons were being passed around from complex to complex, protons were often being pumped from the stroma to the lumen. So in this process of the water splitting, we had a proton in the lumen, and in the process of passing the electron from plastiquinone to cytochrome B6F, transferred, sorry, a proton, right? So altogether, throughout these steps, you're basically accumulating a high concentration of protons in the lumen, which makes it more acidic by definition. And then it also makes the stroma side of the membrane very proton poor, meaning that it was basic. So now you've created a proton gradient, and so we can use that proton gradient to our advantage. So essentially what ATP synthase does is it takes those protons and it allows them to travel up through the barrel. And when it does this, there's like a little bowl on the other end, and that bowl has lots of ADPs and lots of phosphates in it. But all that's needed is some sort of energy to force the bowl to kind of there's almost like a stick that's in somewhere here in the barrel, and that stick can kind of mash together ADP and phosphate in the bowl to create ATP. So what you would need to power that is obviously energy, and you get that by dissipating that gradient. So we allow one proton to travel, the stick, you can call it like let's just call it a wooden spoon. It makes it easier in my mind to understand, but the wooden spoon type of thing moves around in the bowl and it mashes together some of that ADP and that phosphate. And basically with every proton that travels to the other side of the membrane to dissipate that gradient a little bit, the energy that's extracted from that is used to mix together the ADP and the phosphate. And that gives you ATP. So again, just as a refresher, the actual product that we would get from the light reactions is oxygen, and we got that in the water splitting step. But the intermediate products that we get, which are the ones that are used to fuel the Calvin cycle, are NADPH and ATP. So do you guys have any questions about the light reactions so far before we head into the dark ones? Just letting you know, the light reactions are a lot more complex than the dark ones, so it does get a bit easier if this is confusing to you. But I'm just going to check Teams on my phone just to be sure that no one has any questions. Okay, perfect. So it doesn't look like it. So yeah, now we've done the light reactions. So the next step would be the dark reactions, also known as the Calvin cycle. So the goals of the Calvin cycle, the main goal, which produces the only actual product, is to reduce carbon dioxide to synthesize sugars, right? But then one of the intermediate products are to make ADP and inorganic phosphate and NADP, because those two intermediate products are going to immediately be transferred over to the thylakoid membrane to power the light reactions of photosynthesis. So that's kind of what I was showing you here with this diagram. The light reactions, the only real product we got was oxygen, but then we also made ATP and NADPH so that we can use those to power the Calvin cycle. And now that we're going to talk about the Calvin cycle, the only real product that we're producing is sugars, but we also make NADP plus and ADP and inorganic phosphate so that we can use those to power the light reactions. So these never actually leave this cycle. Right, so yeah, and this happens in the stroma, which is again that fluid of the chloroplast. So yeah, looking at the Calvin cycle, it's super, super complicated, but thankfully you really only need to understand the 3 basic steps of the Calvin cycle, and it's not too bad. And of course, considering the fact that it's an open notes exam, hopefully you don't have to do too much memorization, which is nice. But yeah, essentially it comes down to 3 main steps. The first step is called carbon fixation. The second step would be where you reduce 3-PGA to G3P. PGA stands for phosphoglyceracetate, I believe, and then G3P stands for glyceraldehyde 3-phosphate. And then the last step would be restoring RUBP, which is called Rubisco. So let's look at what these actually mean, right? So the first step I said was called carbon fixation. So what happens is carbon dioxide is going to combine with RUBP, which is, I believe that's actually ribulose 1,5-bisphosphate. Yeah, I think that's what it's called. Yeah, so RUBP is ribulose 1,5-bisphosphate, okay? And so the carbon dioxide is going to combine with this ribulose 1,5-bisphosphate to produce 3-phosphoglyceracetate. You're probably like, what does this even mean? What are these molecules? Some of these you don't really need to know too well. Like, you don't actually need to know what this is. Like, you just kind of need to recognize the name. But the carbon dioxide comes from the fact that carbon dioxide is one of the reactants in the whole process of photosynthesis. So I went back to that first slide where it had like the chemical formula for the chemical reaction, for photosynthesis, you would see that carbon dioxide was one of the reactants. And this is where carbon dioxide comes in. It's needed to combine with ribulose 1,5-bisphosphate to produce 3-PGA. So that's what happens in this step. We have our carbon dioxide from the atmosphere, right? And then it's going to combine with RUBP to produce 3-PGA. And that's what we're getting here. So this is 3-PGA. Next step is to reduce the 3-PGA that we made in that first step. So to reduce it, essentially what we're doing is we use ATP and NADPH to reduce that 3-PGA, and that creates glyceraldehyde 3-phosphate, right? Which is G3P. So I guess something that it would be beneficial for you to know is that, again, just as a refresher, reduction means to add electrons to something, right? So if we're reducing 3-PGA, that means we must be adding electrons to it. But where do we get those electrons? Well, we get it from NADPH, because if you remember when we talked about the light reactions, we said that we have in that last step, photosystem 1 is transferring that excited electron over to NADP, which is the terminal electron acceptor. And when it does this, it's basically reducing NADP to create NADPH. And so when this happens, NADP, you can think of it as being sort of an empty suitcase. There's nothing in it. NADPH is more so like it's more so like a suitcase that has electrons in it. So it's kind of full. So what do we use those electrons for? Well, that's the point. We bring it over here to the Calvin cycle, and then we can use those electrons by basically emptying out the NADPH or oxidizing it. And when we oxidize NADPH, we're reducing the 3-PGA to G3P. Because as I said before, when we were talking about cellular respiration, every reduction has to be paired with an oxidation. So if you're reducing something, something else has to be oxidized. So in this case, we're reducing 3-PGA. So we're getting those electrons or oxidizing NADPH to reduce 3-PGA. And when we do that, we produce something called G3P, which is glyceraldehyde 3-phosphate. And then the next step, so when we do this step, actually, what we're doing is part of this process, which you don't need to know exactly how at all, you just really need to understand the idea, is that you're also producing sugars. And like usually it's glucose, but it could be other organic molecules, but more times than not, it's glucose, right? So the way that we do this, you don't need to know, but you're kind of using the glyceraldehyde 3-phosphate to make some sort of sugar. That's really all you need to know, right? So that's something that happens after this step. So why is there still another step if we know that the only product, the real product that we're making from the Calvin cycle is sugar? Well, there's another step because it's a cycle. So for it to be a cycle, it has to keep going round and round and round. So we want to be able to regenerate all of the ingredients that were needed to start step 1 so that we can keep the cycle going. Because right now we basically used everything that we needed. Now we have to regenerate it again so that step 1 can happen again, right? So you still need the same reactants each time. So we need to be able to make sure that we replenish the amount of those reactants so that the cycle can continue. So that's exactly the point of step 3. We're restoring ribulose bisphosphate. So that's what we're doing here. So when we're restoring ribulose bisphosphate, some of the G3P from step 2 is used to make sugars. So that's what you see here. Some of that G3P we're going to use to make our main product, which is glucose and other sugars. The rest of that G3P is going to be used for the purposes of keeping the cycle going. So it's going to be used to restore one of the first reactants, which is the ribulose 1,5-bisphosphate, right? So when we do this, you don't even need to know how it happens. You just need to know that we use some of the G3P to regenerate RUBP. And then the cycle can restart because now we have all the ingredients that we needed. Because if you go back again to step 1, the carbon fixation step, you can see that here carbon dioxide is going to combine with ribulose 1,5-bisphosphate. So those are the 2 ingredients that you need, or the 2 reactants that you need. Carbon dioxide comes from the atmosphere. It diffuses into the leaf or the chloroplast, so that's fine. But the ribulose 1,5-bisphosphate needs to be made by the chloroplast itself. So it needs to regenerate that. And the way we do that is using some of that leftover G3P from step 2. You don't need to know how we do it. You just need to know that it happens. So yeah, that's really all you need to know about the Calvin cycle from my understanding. This was sufficient enough for me in first year, and it was sufficient enough, every year that I've been doing PAL. So hopefully, I don't see why he would test it differently. This is basically the extent of the details you need to know. It's a lot of really weird acronyms, like a lot of abbreviations for some of the molecules. Memorizing it is really all you can do, but also just kind of understanding the cyclical nature of the cycle itself. and the fact that one of the steps is just exclusively dedicated to making sure that the cycle can restart, recognizing that our only actual product is glucose and other sugars, but we make two intermediate products, which are the NADP, which would be the empty suitcase. We make that because when we're reducing 3-PGA, we take out the electrons from the NADPH, so we oxidize it, and then the oxidized form is used again in the light reactions later. And then the other thing that we return back to the light reactions would be ADP, because we use ATP throughout the cycle. And so when we do that, we're basically removing a phosphate and we get ADP. So the ADP goes back to the light reactions so that we can keep those light reactions going, because they also use ADP, right? And then, of course, when the light reactions are done again, we spit back the other reactants that we use specifically for the Calvin cycle. So then it'll give back some NADPH, and then it'll give back some ATP. And then it kind of keeps going. So they basically keep passing these reactants in different versions back and forth. And that can keep the two cycles going. But the only two real products are the oxygen and the sugars. Yeah, this is also the same video that I showed before, but this one has a different timestamp. And obviously the numbers, this is very specific in terms of the numbers of each molecule and stuff, and the specific reactions that happen in the Calvin cycle. That's not really important for your purposes. You will be more than fine without it. You don't need to worry about that at all. It just is like a nice animation, I think. And when I'm studying, I really like watching like videos that put everything together while I read the notes, because then I feel like it's a lot easier to memorize that way and a lot easier to understand. Yeah, so we can move on to signal transduction, but I just want to make sure that everyone is okay with the things that we've done so far. I'm just going to check the Teams chat just to be sure. It doesn't look like there are any questions as of now, but yeah, does anyone have any questions? I have to run in a little bit, but because my computer was being incredibly annoying, I'm going to try to stay for a bit longer. If you guys have to go, obviously feel free to go. The session is like technically over, but I will just go through this last one. It does take quite a while. Would you guys rather I go over the entire thing? Would you guys rather I go over part of it, or would you rather I just keep it on D2L and then I'll go over it next week? It's up to you. I did go over it on my Tuesday session. Okay, next week. Okay, sounds good. Thank you guys so much for joining. I really appreciate you bearing with me during my technological issues. I wish my computer wasn't like that, but I'm just gonna probably have to update it, I guess. It's probably why it's lagging so much. But yeah, thank you so much for coming, and I'll see you guys next week. Have a great weekend.